Hello everyone. Good evening to all of you. Please confirm if you can see the screen and if you can hear me. This is the last session from my side. Of course, 2 more days to go where you will come across other 2 units. How is your preparation going on since the exam is very near, right? Thank you, Harshita, for confirming. So, how is your exam going on? How is your preparation going on for all the units? I guess it should be at highest peak because we are left only with very few countable days. And at this stage, you should always focus on doing more and revision and practicing the questions, right? Okay, so in my previous session, that is in the last week, I have taken few questions from applied biology. Today, I'm just taking up rest of the questions. So have a look. As I told you, agrobacterium is considered to be one of the important topic when it comes to Unit 12. So here is a question for you. There is a schematic representation of tDNA region of a transgene construct. Okay. And the southern blot analysis. This is the southern blot analysis. This is the transgene construct. So you will find 5 independent transgenic lines are there that is labeled as A to E. So here you are provided with restriction enzyme for digesting the genomic DNA. You are getting 5 independent transgenic lines that are developed using the construct. Then you will find a probe that is used for hybridization, which is seen as a black box below. So this is your probe. Now, from the given image or from the given analytical data, you have to identify what is the correct list of single insertion transgenic events. Single insertion transgenic events. Have a look and see and tell the correct answer over here. As I always say, the image-based questions are not so tough. It is easy to answer. 2 things you have to focus on. You have to read the question thoroughly, and at the same time, you have to analyze the image thoroughly. So that is what is utmost important. So that is not only applicable for Unit 12, it is applicable for every unit, whether it is your Unit 4, Unit 10, where the graphical representations are more, then Unit 1, right? So wherever you come across this image or graphical representations, try to attend that question because majority of times the answer is there in the question itself. Yeah. Okay, Harshita, I can see your answer. You have given as option 2 answer. What about others? What about others? What about others? Good evening, spiritual science. So, do you have any answer for this question? So, a transgene is there, a probe is there, restriction digestion done. Then you have to analyze the blot. Blot analysis, you have to identify the correct answer. Any answer from your side? Any answer from anyone else? No? You can get into the answer. Try out everyone. See, now the exam is also near. And you, as I told you in the beginning, you should start practicing more and more questions. This is the stage where you have to keep on practicing the questions. So Brahmastra series is one such opportunity where you are getting unit-wise question discussion, along with the corresponding explanation as well. So take this opportunity and interact and give the answers. So the correct answer over here is, as you mentioned, yes, it is option 2. Now, why option 2 is the correct answer? Any justification for this? Why C and D are eliminated from here? C and D are eliminated from here. Any specific reason behind this? So when you go for genomic restriction digestion with the help of PST1, you will find 3 fragments, right? 3 fragments you are getting, of which you can find that the probe is getting attached to the first 2 fragments but not to the third, right? You can see through the image. So in that case, what you can predict? UT is present for all the transgenic lines. There is no difference in that. But wherever you get extra 2 bands, that indicates your single insertion events, because the probe is not able to bind to the third fragment. So you cannot use that. So first 2 you can. So in that case, you should get 2 bands. So in that aspect, if you analyze the blot of A, blot of B, and blot of E, apart from UT, you are getting only 2 extra bands, right? As per our experiment. But if you analyze the blot for C and D, you are getting more than 2. So here you can see 1, 2, 3, 4, 5. Here you are getting 4. So that's the reason C and D are eliminated from the given options. See, given the transgenic construct contains a tDNA region with restriction sites for PST1, and the southern blot analysis was performed using PST1 digested genomic DNA from 5 transgenic lines, that is A to E. A probe was given to identify the transgene integration. So from the probe, these are the following observations. The probe can bind only to the 2 fragments after the digestion, which is marked as 1 and 2. So here I have marked as 1 and 2. Right. After that, the third fragment does not hybridize with the probe, so you cannot get the result within the blot. Next, UT lane will remain as a single band and it is common for all the samples. UT is the untransformed one. So the transgenic lines should display the 2 additional bands which will be corresponding to probe binding fragments. So that will indicate a single insertion event. So from this observation, what we can see, lines A, B and C, E exactly shows the 2 bands conforming a single transgenic events. Whereas for the lane C and D, there are 2 additional bands which indicates multiple insertion events. And that's the reason we have eliminated C and D. Okay. Moving on to our next question. Now this is related to plant tissue culture. Friable callus provides the inoculum to form cell suspension cultures. The process known as habituation refers to the property of all callus that loses the requirement of auxin and cytokinin during long-term culture. Cellulase and pectinase enzymes are used for generating protoplast culture. During somatic embryonic development, torpedo stage embryo will be formed before the heart stage embryo. Now here also you have to identify the correct combination of statements. Identify. So basically when it comes to plant tissue culture, it comes in the form of a combination. Like if you have some idea about unit 6 plant physiology, that information will help you to incorporate in this question as well, like tissue culture related questions as well. Like you have got the plant hormones, auxin, cytokinin, then protoplast, embryogenic development, which you might be studying, you might have studied under Unit 5, plant developmental biology. So basically, this becomes a combination of the units which you have already studied. Yes, what should be the correct answer over here? Now, at least this I should get an answer from everyone. What is a callus? Can anyone tell me what is a callus is all about? Or how do you define callus? How do you define callus? See, if you are not interacting, then how could you realize that what you know or what you don't know, and how could I can understand that you are able to understand or not? Yes. So whoever is seeing live, try to interact, students. So this is not a one-way road, this is a two-way road. So both of us can interact. You are giving us the answers. So we are helping you to dive into the correct answers in case if you are giving an incorrect answer. And this is a very simple question again. So never avoid Unit 12, because there was a time when we used to say Unit 12 is not so important from examination point of view. But in the recent past, you can see how many 4 marks questions you are getting from Unit 12. So it's like year by year, even the ignored topics are gaining the importance. What should be the correct answer for this question regarding plant tissue culture? What is a callus? What is a callus all about? It's an undifferentiated mass of cells, you might have heard, right? Which is continuously proliferating, but it is not undergoing differentiation. And what hormones are required for maintaining the callus? We have auxin and cytokinin. And what is the concentration for that? Almost equal, because if auxin increases, it results in root formation, and if cytokinin increases, it results in shoot formation. So when you are giving them in equal quantity, it is at that stage we call callus has been generated. So the correct answer over here is statements A, B, and C. So why C is correct? You know what is a protoplast is? Have you heard about protoplast, anyone? Have you heard about protoplast, anyone? These are the cells without cell wall. And cell wall is made up of what? Cellulose and pectin, right? So when you are giving the cellulase and pectinase enzyme, they will be degrading the cell wall. So what you are getting a protoplast out of that. And why the statement D is incorrect? Let's have a look onto that. So you have distinct stages of development when it comes to somatic embryogenesis. So what was the statement D? During somatic embryo development, torpedo stage embryo will be formed before the heart stage embryo. So this is incorrect, because torpedo stage is formed after the heart stage. So you have different stages of development: globular stage, heart stage, torpedo stage, and cotyledon stage. So before the heart stage, we have globular stage. So it starts with a single cell, then it forms a group of cells. The group of cells accommodate together, forming a globular stage. Then it enters into a heart shaped embryos, and finally the torpedo stage, and from there you'll get your cotyledon. That is the true leaves. So these are the four important stages of development. Next, so what are the significance of each stage of development? If you see, for globular stage, the embryos will look small and round, and by nature they will be multicellular. In the heart stage, they will have bilateral symmetry. That means, as the name indicates, the shape will changes to heart, but it will have more cotyledon development. Next, what you are having, torpedo stage. In torpedo stage, what happens? It consists of the initial cells that are required for the development of shoot apical meristem and root apical meristem. And finally, the cotyledon stage, that is the mature stage, you will find that the embryos look cylindrical in structure. So that is what the last stage. So you have to remember the sequence of different stages of development, and at the same time, significance of each stage. What is happening on each stage? Now, you can classify the embryogenesis into two types. First one is the direct embryogenesis. As the name indicates, it involves the development of embryos directly from the cells of explant. That means if you are taking the immature embryos as your explant, so from there itself the development takes place. It does not undergo any intermediate stages like callus formation, and that's the reason we call them as pre-embryogenic determined cell, because their fate is already determined what they are going to be. So we call direct embryogenesis stage as pre-embryogenic determined cells. The second one is indirect embryogenesis. Now, this involves the formation of somatic embryos, which undergoes repeating cycles of cell division, and it also have an intermediate stage where callus growth takes place. So, overall speaking, indirect embryogenesis is a multi-step process where the development does not take from the explant. It undergoes cycles of division, callus growth, and then you are getting the corresponding development. So cells that are not carrying this pre-embryogenic determined cells, they are induced to differentiate and form the embryos by exposure to several suitable treatments. So these indirect embryogenesis, what they will do, they will provide them with some chemical treatments, after which the cells will be transformed into pre-embryogenic determined cells. So since you are injecting or inducing them to become the stage, we call this indirect embryogenesis as induced embryogenic predetermined cells. Okay, so be clear with the terminologies. Direct embryogenesis is related to pre-embryogenic determined cells. Because there is no intermediate stage, direct development takes place. Indirect embryogenesis is a multi-step process where you are inducing it to undergo processes in such a way that it develop the pre-embryogenic determined cell. So that's the reason we are inducing, we call it as induced embryogenic predetermined cells. Okay. Moving on to our next question. So this is a very easy question. At least I should expect some answers for this. Match the following. As you can see in the column A, you have auxin, protoplast culture, cytokinin, microspore culture. Column B, we have embryogenesis, 6-furfuryl aminopurin, pectinase and cellulase, and then we have indole 3 acetic acid. What should be the correct combination over here? Okay, Nagashri, I can see your answer. You have given 2 as the answer. What about others? What about others? Come on, this is very easy question. What is an auxin? What is a cytokinin? Protoplast, just few slides back only I had mentioned what is the protoplast is all about. You can easily correlate to the column B, that is their corresponding options or the combinations. What is the precursor of auxin? Who is the precursor of auxin? What is cytokinin related to? What is the protoplast? If you know these basic things, you can easily get a 4 marks out of this question. But the problem is that you are not responding to this basic questions. So I'm quite unsure whether you are knowing it or unknown. I'm not sure. Absolutely correct, Nagashri. So the tryptophan will be the precursor for auxin, whose structure will be similar to your, that is the indole ring will be there in their structure, right? So that's why you can easily mention that auxin is related to indole 3 acetic acid. So once you get A to 4, you can also choose elimination pattern. So you can eliminate option 1 and option 3. So now what is left? 2 and 4. Next, just few slides back only I told you protoplast means they does not have cell wall. So if you want to remove the cell wall, what treatment you have to give them? Pectinase and cellulase. So that will degrade away the cellulose. Sorry, the cell wall. So B to 3. Now you got your answer under your confidence. Now just randomly have a check what about others. Cytokinin, these are adenine derivatives. So 6-furfuryl aminopurine. Microspore culture is commonly used for embryogenesis. So like that, you can easily eliminate the options and get into an answer. Okay. So this is just a reminder that we have already started with AIML, that is bioinformatics, computational biology, where you will get the opportunities to do project work for about 3 months, 6 months, or even 12 months. Paper publication assistance will also be given to you. So under this, you can work on AIML in drug discovery projects, cancer and disease prediction, protein structure prediction, genomics, transcriptomics, and nowadays you know that the trend is going on for AIML, bioinformatics. So this holds a very good opportunity to make your profile better for the upcoming times. So have a look onto this, and you can always reach us to this through this call toll free number that is being displayed in the screen. Okay, so if you want to know further assistance, if you want to know more information, you can always call us in this number. Remember, both online and offline project works are also going on. Moving on to our next question. Again, this is also related to plant tissue culture with respect to hormones only. A transgenic tobacco plant overexpressing IPT, that is isopentenyl transferase, under the control of a promoter region of senescence associated receptor kinase, we are exposed to drought conditions for 15 days, followed by rewatering for 7 days. So, the following hypothesis we are proposed regarding the changes in the transgenic plants, which you can see at the end of 7 days of rewatering. The plants would be wilted and fails to survive. The plants would be healthy and survive. The plants would show higher proportion of cytokinin compared to wild type plants, or they will show higher production of abscisic acid compared to the wild type plants. Identify the correct combination of hypotheses from the given statements. Identify the correct combination of hypotheses from the given statements. So if you have an idea about cytokinin, drought, senescence, these are some terminologies which you can have a look with respect to different plant hormones. Different means here they are only asking about cytokinin and abscisic acid. So, based on that, you can get into a conclusion for this question. Can I get some answers for this question? So, do you feel plant tissue culture tough? Because I'm not getting any answer for this. This is the third question related to plant tissue culture. Once upon a time, they used to ask a lot of questions from this plant tissue culture part. And the good fact about this topic is that you don't have to put extra efforts because, anyways, you are studying plant physiology, that is, Unit 6 in very much details. So, in that aspect, you just have to apply the information from different hormones and over the tissue culture laboratory, whatever the work is being carried out. So what should be the correct answer over here? Harshita, Nagashri, spiritual science, any one of you? The correct answer over here is a combination of statements B and C. That is, the plant would remain healthy and survive, and the plants will show higher production of cytokinin compared to wild type plants. So, why statement A and D are eliminated? Obviously, if the statement B is correct, you can eliminate statement A. So, after rewatering, the plants are able to survive. So, obviously, if statement C is correct, after rewatering, at that junction, you will find high production of cytokinin will be there. So, cytokinin will induce what? Delay senescence. So, you can get into a conclusion statement B and C are correct. The points to be remembered is IPT, that is your isopentenyl transferase enzyme, is involved in cytokinin biosynthesis. Next, you have got PSARC, that is the promoter region of senescence associated receptor kinase, which is activated under stress conditions like drought, flood, and all. So, if you combine this IPT and SARK together, so overexpression of this IPT gene under this promoter will obviously lead to overproduction of cytokinin, which induces delaying the senescence, thereby promoting the stress tolerance. So, these are the features or functions of cytokinin. So, the plants would be wilted and fail to survive is definitely incorrect, because if higher cytokinin levels are there, then it is going to improve the drought tolerance. So, that will delay wilting and promoting recovery after rewatering. So, that is the main part, because if you remember, if you have studied, cytokinin is the plant hormone which helps in delaying the leaf senescence. Next, while the plants should show higher production of abscisic acid compared to wild type plant is incorrect, because ABA is a kind of natural plant hormone that gets activated during the drought condition. But transgenic plants with higher cytokinin levels will tend to lower the ABA levels, because these two hormones are antagonistic to each other when it comes to stress responses. Right? So that's the reason statement D is also incorrect over here. Moving on to our next question. Following genes have been genetically engineered to develop herbicide resistance in plants. Resistance to glyphosate using 5-EPSP, that is 5-enol pyruvate shikimate 3-phosphate synthase gene. Bilaphos resistance using the BAR gene. Sulfonyl urea resistance using the acetolactate synthase gene, and atrazine resistance using the glutathione S transferase gene. So you have to identify two cases of mechanism that is based on abolition of herbicide binding to the enzyme. The correct combination over here. What is the correct combination of statements over here? For this, you have got a table that will give you some insights about different marker genes. So, if you remember the table, you can easily get into a conclusion. Can I expect some answers? So, the correct answer over here is option 4, that is statement A and statement C are correct. So, why statement A and C are correct? Have a look onto this. As I told you, we have got a table that will describe you with some categories of marker genes. As you can see, we have antibiotic resistance genes that includes your NPT2, NPT1, HPT, bleomycin resistance, and then aminoglycoside adenyl transferase. So NPT2, the substrate used for selection will be kanamycin and geneticin. For for NPT3, you have got the same substrate. For HPT gene, as the name indicates, hygromycin. For bleomycin, as the name indicates, bleomycin substrate. And for aminoglycoside, the substrate used for selection will be streptomycin and spectinomycin. Second category is anti-metabolite markers. That includes dihydrofolate reductase, the most commonly used marker DHFR, and the substrate used over here is methotrexate obtained from mouse. So here majority of them are E. coli with two microorganisms somewhere, Streptococcus and Shigella. Then we have DHPS, which is using sulfonamides as their substrate for selection. Next, we have herbicide resistance gene. So here you come across BAR, EPSPS, then you have got the bromoxylane, glyphosate. So these are the examples of herbicide resistance gene. So for BAR, you can see it is here glucosinate L-phosphoinothricin. For EPSPS, glyphosate. Then you have got acetolactase, glyphosate, oxidoreductase, and bromoxylane nitrulase. Other than this, we have got another category, we call it as others, where you will come across beta glucuronidase enzyme. Then you have got xylose isomerase enzyme. mannose 6-phosphate isomerase, and betaine aldehyde dehydrogenase enzyme. So, depending upon the marker genes, you will have the source and you will have the substrates that are being used for the selection. So, majority of cases, you will find that the substrate that is being used for selection is the term somewhere related to the gene itself or the enzyme itself. Of course, not for everyone, but yeah, to some cases you can. Okay. So this table is important when it comes to examination point of view. In any aspect, you can get a question in the form of statements that I have shown you, or maybe in the form of match the following. And this is just a summary of some important transgenic plants with their applications. So if you see, we have BT cotton. Why do we have BT cotton? Because these are pest resistance, herbicide tolerance, provides better yield, and the most important function is they are resistant to bollworm function infestations. Wheat, they are resistant against herbicide. Brassica napus, a gene which encodes hirudine. What is hirudine? A protein that prevents blood clotting. These are being chemically synthesized and transferred to Brassica napus. So, this gene will be activated in the plant and start synthesizing the protein which accumulates in the seed. Now, you can extract and purify the corresponding product and can be used for as a medicine because it prevents blood clotting. Tobacco, we have CPT1 gene introduced that will show resistance against pest. Flavor saver tomato, you already have an idea which involves delay in ripening, and polygalacturonase enzyme is being used. Golden rice, we have it vitamin A rich. Potato, this is they are generating transgenic potato for higher protein content. Bananas, transgenic bananas, they act as edible vaccines to protect children against diarrhea. When we have transgenic soybean and maize, which can be used because they have herbicide resistance. Okay, so these are the different applications of transgenic plants, some of the important transgenic plants. Moving on to our next question. Now, this is related to several secondary metabolites and their medicinal uses. Okay, so you have to identify the correct match among them. So, these are being used in traditional medicine across the world. So, many of these compounds can be purified or synthesized and are used in modern medicine. So, you have the metabolite name, you have got the plant source, and then you have got the medicine or the use. Identify the correct combination over here. This is also again part of secondary metabolites from Unit 6, right? Where you will study about secondary metabolites, but it also comes under the category of Unit 12, where like bioresources and biodiversity. I think it's 12E. So that can be considered under this category. Okay, Nagashri, any other answers? Any other answers? So for this also we have a table. If you can remember that, it is going to help you out. And this was, I think it was asked recently, not immediate recently, but yeah, within the 5 years, you have got this question. So, according to the recent trend, you can always expect unexpected, we say, right? So, you can expect from any part of Unit 12, and that too experimental, and at the same time 4 marks analytical question. So you have to think twice before skipping this unit. Of course, once upon a time we used to say this is not so important, we can skip this. But now we cannot say this part because this is also playing a very important role in our CSIR NET exam as well. Digoxin, salicin, morphine, artemisinin. What should be the correct answer? Can I expect any answers other than Nagashri? Anyone else? So the correct answer, as you mentioned, is correct, Nagashri. It is option 4. So here is a table for you where the drugs name are given, what are their sources and what is their uses. There are probably there could be a problem in remembering what is the source over here, but you should always focus on what is drug and what is its use. So atropine, it is considered to be a relaxant or we can say a sedative. Codeine, it's a painkiller. Colchicine, it's an anti-cancer agent. Digitoxin, it's a cardiac stimulant. In fact, the source is also digitalis. So you can remember in that way. Then we have got menthol, obtained from mint. Nasal congestion, used against. Morphine, again, it's a painkiller. Quinine, it is used to overcome malaria. Reserpine, hypertension. Scopolamine, it's a kind of sedative. Then we have got the anti-cancer drugs like Taxol, vinblastine, and vincristine. So these are anti-cancer and these are helping in curing leukemia. Okay. So if you remember this kind of table, it will be easy for you to get into an answer. So this is the same thing, but you will have some extra information also in this table. So you have artemisinin, anti-malarial, azadiractine, insecticidal, capsaicin, rheumatic pain treatment. Then similarly, you have got chiconine. Again, it is antimicrobial. It is a red pigment which is used in cosmetics like lipstick and even dyes for silk. Okay. And as I told you, these two are anti-cancer agents. Okay. So the like this you can get different examples. Based on that, tissue culture experiments are being carried out. Okay. Moving on to our next question, that is regarding gene therapy, another important topic from examination point of view. So it's a promising tool for addressing several diseases in humans. So based on that, you have to identify the statement which is false. Gene therapy involves the direct modification of the cells or the model to achieve a therapeutic goal. Current gene therapy is directed at modifying somatic cells. The only successful gene therapies are those in which cells are removed from a patient, genetically modified, and then reintroduced back to the patient. Recessively inherited disorders are good targets for gene therapy. So here you need to identify the incorrect answer. So always when you read the question, make sure that you are reading it properly, whether they are asking you correct, incorrect, that you have to be sure of. I hope so you all are aware about gene therapy, what are the approaches, what are the types, all those things you are aware. So this is also one trending topic when it comes to Unit 12. Okay, Nagashri. So you have given option 1 as the answer. What about others? Is there any other answer? Here they are asking you the incorrect statement, false statement, true, not true, false, incorrect, not correct. These are some terminologies which you should be keen on, or you should be alert on while reading out the question. Because when we are in the examination hall, sometimes we see a question that we are already aware. Like, for example, I just saw a question mentioning gene therapy. So there are chances we become excited and we jump into an answer. So that should not be done. Have the patience, read it thoroughly, and then jump into the answer. Okay, you have opted for option 2. Okay. So, what about others, participants who are listening to this lecture? Try to give the answer. There is nothing to worry about giving a right or wrong answer. All you need to do is give the answer so that you can understand what is inbuilt in you, where you are moving on, how much confident you are with this topics. Very good participation, Nagashri. So let's have a look into the answer. What should be the correct answer? The correct answer over here is statement 3. Now, why statement 3 is incorrect? Because this is not the only gene therapy. We have two different types. First is you can classify the gene therapy based upon the type of cell. That is somatic cell gene therapy. Another one is germline gene therapy. So, as the name indicates, here therapeutic genes will be transferred into the somatic cells like bone marrow, blood cells, skin cells, etc. And since it is somatic, it will not be inherited to later generation. So, at present, all the researchers are directed to correct the defects in somatic cells. On the other hand, as the name indicates, the therapeutic genes will be transferred to the germ cells, and we have only two, that is, eggs and sperms. And since we are transferring it to the germ cells, so obviously these are heritable and passed on to upcoming generation. So, for safety, ethical, and technical reasons, it is attempted at present. So, these are types of gene therapy based upon the cell type. Now, if you see, we have two approaches of gene therapy which are successful. One is direct delivery method, another one is cell-based delivery method. So, in direct delivery method is also known as in vivo gene therapy, where we are inserting the therapeutic gene into a vector, and that vector could be a vehicle, that is a virus, any virus, which is then injected into the patient that will be targeting the specific organ. Another one is the cell based delivery, or we can also call it as an ex vivo gene therapy. In this case, the genes, first, the stem cells will be isolated from the patient itself, grown in the lab. You will carry the therapeutic gene in a vector, and you will combine this together. So, what you will get? Genetically modified cells you will be getting. So, they will undergo multiplication under in vitro condition. After the multiplication, they are being re-administered back to the patient. So that is what it is mentioned, the third point. So, but it is successful. At the same time, this is also successful. Both the approaches are applicable. Okay. So this is yet another image if you need more clarity. So direct gene delivery, we are taking the therapeutic gene, incorporating them into a vector. Vector could be a virus like adenovirus. Then they are directly given to the patient body, or they are directly injected into the patient body. So they will reach the target organ. Cell-based gene delivery method means we have got therapeutic gene that is incorporated into the vector. So that will be a harmless virus like retrovirus and lentivirus. Meanwhile, we have isolated the stem cells and growing them in the lab. Now, this corresponding vector will be introduced into the stem cells. So, togetherly, they will be multiplied and now they will be reintroduced back to the patient. So, that is, we call it as a cell based gene delivery system. Okay. So, both are successful. Of course, they have their own set of pros and cons. Moving on to our next question. A pharmacy student designed a drug to specifically target the receptors for retinoic acid in order to prevent stem cell differentiation. So, after the in vitro trial, the investigator found that the cells underwent differentiation and the drug seems to be ineffective. So, here some reasons are given by the student. First, the size of the drug exceeded the size of the molecules that could cross the membrane. Second, the drug was small in size but hydrophobic in nature. And third, the drug did not bind to its receptor. Now, you have to identify the most probable reason for the drug ineffectiveness. Identify. I hope so you got it, Nagashri, why the statement 3 was correct and why not 1 and 2. So what should so this is this kind of question does not require you should study something and come to the or attend this kind of question. This is a kind of logical question. Think about the question, what is this drug doing so? And you have to apply your analytical skill to get into an answer, because this no one can explain you what it comes under which category. It is something related to drugs, right? The vaccines, which is there in your unit 12. So these kind of questions you yourself should analyze and get into a conclusion. What should be the correct answer for this question? No answer? Very simple is this question. See, what they have mentioned is a pharmacy student designed a drug specifically targeting the receptors for retinoic acid in order to prevent stem cell differentiation. So after the in vitro trial, what they found that cells underwent differentiation and the drug seemed to be ineffective. So what is the reason of ineffectiveness? So you can correlate the statement 1 and 3. The drug did not bind to its receptor. What could be the reason for that? Because the size of the drug exceeded the size of the molecules that could cross the membrane. Simple. So you can get the answer from the question itself directly. This cannot be taken into consideration, but these two are correlated with each other. Right, because whenever a ligand goes and binds to the receptor, there should be an affinity, there should be a complementarity between them. So if these two things are not there, then it won't be internalized or it won't be taken up inside. So this is how you need to predict the answer for these kind of questions. Now, yet another question related to vaccines, and this is a recent question. Following are some statements made about vaccines. Covaxin is a killed cell vaccine. Corbevax is a subunit vaccine. Oral polio vaccine is given as a live attenuated vaccine to adults and as a killed vaccine to children. Third generation vaccines against smallpox were based on attenuated vaccinia virus. MMR vaccine is given to children to protect them from diphtheria. Now, here you have to identify the correct combination of statements. So, this is a general knowledge question, I can say. These are the things which you see in news channels, right? So this was last year. This question was asked. In fact, 2 or 3 questions were asked based on this Covaxin and all COVID vaccine. So whenever you are studying about vaccines, always try to find out the recent trend. So that will be more fruitful for you. Because see, this is a 4 marks question. You have to be very clear with the types of vaccine also. So that becomes the part of Unit 4 as well as Unit 12 from your CSIR NET Life Science syllabus. What is the answer for this question? Are you all aware about different types of vaccines? Yes or no? See, the correct answer over here is A and C only. So, why A and C are the only correct options? Let's have a look. Covaxin is an inactivated virus vaccine, and Corbevax is a subunit vaccine. Subunit means a protein or a sugar is attached. So, here you are getting statement A as correct. Second, the oral polio vaccine is a live attenuated vaccine and is typically given to children, not to adults. Similarly, the inactivated polio vaccine is given by injection and it can be administered to both children as well as adult. So that's why statement B is incorrect. Then, third generation smallpox vaccines like modified vaccinia ankara are based on an attenuated form of vaccinia vaccine. So, statement C is also correct. Then, MMR vaccine protects against measles, mumps, rubella, and not diphtheria. So, diphtheria is covered by the DTP, diphtheria tetanus pertussis toxin, sorry, vaccine. So that's why statement D is also incorrect over here. Okay. Now, Hanji, just giving you some glimpses of different types of vaccine. So you can classify the vaccine based upon whole organisms and purified macromolecules. Live attenuated vaccines, these are the diseases which can be cured. Advantage is that they generate stronger immune response, often lifelong immunity with few doses. Disadvantage is that they require refrigeration, storage, and can also mutate to virulent forms. Inactivated or killed vaccine, these are the diseases. These are stable and safer than live vaccine. Refrigeration is not required, but it produces weaker immune response. So booster shots are applicable. Second category is the purified macromolecules, toxoid, diphtheria, tetanus, subunit vaccines. This is also as an example of inactivated exotoxin, hepatitis, pertussis, streptococcal pneumonia. Then conjugate, we have Haemophilus influenza and streptococcal pneumonia. So if you see here, polio sabin vaccine comes under live attenuated. But in this case, polio sock vaccine comes under inactivated or killed. Similarly, hepatitis A vaccine comes under the category of whole organism, inactivated or killed. But hepatitis B vaccine comes under the category of purified macromolecule subunit vaccine. So small things you can have a check on to that. And these are the definitions of different vaccine. So live attenuated vaccines are weakened version of living microbe that cannot cause disease. Inactivated or killed vaccine means the microbes are being killed with some chemical treatment, heat, or radiations. Subunit vaccines means that includes your antigens that best stimulate the immune system. Toxoid, they are formalin inactivated toxins. Conjugate, as the name indicates, they will be conjugated where antigens are linked to sugar molecules. So, sorry. So, previously I was mentioning about subunit like that. No, it is conjugate. Okay. Subunit is just the epitope that can stimulate the immune response. Then DNA vaccine, these are important antigens to be introduced inside the cell. Recombinant vector, here you are using an attenuated virus or a bacterium as a vector to introduce microbial DNA into the target cell. Okay. Okay then, so that's all from today's session. So once again, I'm saying Unit 12 is important, and please go through the Unit 2 important topics. You can expect questions from here both in the form of 4 marks and 2 marks. So days are very less. Keep on practicing more of questions and do more of revision. The crisp notes are important, and wishing you all the best for the upcoming exam. Thank you so much.